Virtually every life process depends on proteins. The functions of proteins depends on their structures. The complexity of protein structure can be best understood at 4 different levels. Primary structure, secondary, tertiary, and quaternary structures. Primary structure refers to sequence of amino acids in a polypeptide. Secondary structure refers to regular arrangement of adjacent amino acids in a polypeptide. Secondary structure is exemplified by alpha helix, beta sheet, and beta turn. Tertiary structure refers to arrangement of secondary structure in a domain and further arrangement of those domains in a polypeptide. Some proteins are made up of 2 or more polypeptides. The arrangement of these polypeptide subunits is called the quaternary structure of the protein. Now, let's go to part 1, primary structure. Here is an example of primary structure. It is a sequence of amino acids of insulin A chains, a very important hormones we are going to encounter for the entire course. The question we ask here is how amino acids are joined together to form a polypeptide. Here are 2 amino acids. The carboxyl group from one amino acid contains with amino group from another amino acid. After eliminating one water molecule, a peptide bond is formed. Now, each amino acid is called amino acid residue. Central carbon is alpha carbon. Further away are beta and gamma carbons. The final product is called dipeptide because it consists of 2 amino acid residues. The end with free amino group is called amino terminus or N-terminus. The one with a free carboxyl group is called carboxyl terminus or C-terminus. For visual learners, formation of a peptide bond can be better represented by using a ball stick model. A peptide bond has 4 characteristics. First, partial double bond characters. As we know from organic chemistry, a double bond is not free to rotate. As a result, the carbon-nitrogen bond is not free to rotate. Second, rigid and planar. The 4 atoms, oxygen, carbon, nitrogen, and hydrogen, are on the same plane. Third, trans configuration. Oxygen and hydrogen are on the opposite sides of carbon-nitrogen bond. Fourth, uncharged but polar. Presence of oxygen and nitrogen make the peptide bond polar, but there's no charge associated with peptide bond. There are several ways to determine primary structure of polypeptide. Most commonly used techniques are peptide sequencing using Edman reagent and DNA sequencing. Now, let's switch to part 2, secondary structure. Secondary structure results from folding of adjacent amino acids into a few regular 3-dimensional forms. Secondary structure include alpha helix, beta sheet, beta turn, and loop. Hydrogen bonds are key to stabilizing these structures. First, let's look at alpha helix. There are 2 types of alpha helix, left-hand alpha helix and right-hand alpha helix. Intrachain hydrogen bond is important in maintaining alpha helix. The example we show here, the carbonyl oxygen is hydrogen bonded to nitrogen in the same back bond, 3 amino acids away. These are the characteristics of alpha helix. In alpha helix, there are about 3.6 residues per turn, and the side chains are facing outward. Alpha helix is disrupted by proline and glycine. Proline is too rigid and glycine is too flexible. Here is an example of alpha helix in myoglobin. In myoglobin, you can easily pick up 7 alpha helix. And as you can see from here, most of them are right-hand alpha helix. Next, we are going to look at the beta sheet. Beta sheet are pleated. It refers to 2 strands come into close contact. Nitrogen and oxygen from the peptide bonds are involved in hydrogen bonding. Beta sheets are typically represented by broad arrows running from N-terminus to C-terminus. Beta sheet can be parallel or anti-parallel. Here are 2 examples of beta sheet. How can we tell a beta sheet is parallel or anti-parallel? First, we look for carbon and nitrogen in a peptide bond. For example, we have 2 panels. On panel A, there are 2 strands. In the left strand, here is a peptide bond. From carbon to nitrogen, the peptide bond pointing downwards. So, the left strands run from top to bottom. On the other hand, the right strand, the peptide bond is pointing up, so the right strand runs from bottom up. Therefore, this beta sheet is anti-parallel. Using the same methods, we can easily figure out the beta sheets on panel B. This is a parallel beta sheet shown using a ball and stick model. Here, oxygen is represented in red and nitrogen in blue. Hydrogen bonds are formed between oxygen and nitrogen from the peptide bonds. This is an anti-parallel beta sheet. Very similarly, hydrogen bonds are formed between oxygen and nitrogen from peptide bonds. Beta sheets typically favors protein aggregation. This has clinical correlation. For example, in patients with Alzheimer's disease, the amyloid protein forms beta sheet and aggregates in the brain to form fiber, similar to silk fiber, which impairs the function of neurons. Second example is Huntington disease, in which Huntington protein aggregates through beta sheet. Alzheimer's disease is a neurodegenerative disease. Key symptoms include loss of memory and thinking skills in patients in their 60s. It is caused by mutation in one of the 3 genes, APP, presenilins 1 and 2. Huntington disease is a progressive brain disorder. Key symptoms include uncontrolled movement. It occurs in patients in their 30s or 40s. It is caused by mutation in the HTT genes. This gene codes for Huntington protein. Prion is a protein virus derived from PRP protein. Everyone has PRP protein, but in prion, this protein is misfolded with expansion of beta sheet. Among diseases caused by prion, Creutzfeldt-Jakob disease, or CJD, is best known. Prion protein can cause other normal PRP protein to misfold and it becomes infectious. Porin is a transmembrane protein found in the outer membrane of bacteria. This protein acts as a channel that transports chemical compound across the membrane. The key features of this protein is that it has extensive beta sheets to form beta barrel. Mutation in these proteins increase the antibiotic resistance of bacteria. Now, let me introduce you the third element of secondary structure, beta turn. Beta turn functions to reverse the direction of polypeptide chain. It includes charged amino acid residues. It is stabilized by hydrogen bonds. And it is usually composed of 4 amino acids, including proline and glycine. As you may recall, proline and glycine disrupt a-helix, but they work well in beta turn. This is an example of beta turn. Hydrogen bond is formed between first and fourth amino acid residues in beta turn. Finally, this is the fourth element of secondary structure, the loop. Loop functions as a linker. It links regular secondary structures. Now, let's switch to part 3, tertiary structure. Tertiary structure is the folding of secondary structures. Based on tertiary structure, proteins can be divided into 2 broad classes: globular proteins and fibrous proteins. Globular proteins have several key features, including spherical shapes and good water solubility. Fibrous proteins, on the other hand, have different features. For example, typically they have a long, narrow rock-like structure, and they have low water solubility. Forces involved in maintaining tertiary structure is an important subject for MBME. These 4 groups of forces play an important role in maintaining tertiary structure. They are hydrogen bonds, disulfide bonds, and hydrophobic interaction, and electrostatic interaction. On the other hand, protein can be denatured. After a protein is denatured, it loses its original tertiary structure. Methods to denature proteins include physical methods and chemical methods. Most proteins do not reverse to their original tertiary structures after denaturation, except ribonuclease. Now, let's switch to quaternary structure. Quaternary structure consists of association of multiple polypeptides by non-covalent interactions. Here is an example of quaternary structure, and this is hemoglobin. It is made up of 4 polypeptides, 2 alpha subunits, and 2 beta subunits. Because subunits are held together by non-covalent interactions, forces in maintaining quaternary structure include 3 forces that I described to you earlier, except disulfide bonds. Clinical correlations of quaternary structure can be represented in sickle cell anemia. This disease is caused by mutation in the beta globin genes. At position 6 of beta chains, glutamic acid is changed to valine. This mutation makes hemoglobin proteins very sticky. Hemoglobin tends to aggregate, which distort red blood cells into a sickle shape. At the low concentration of oxygen, the sickle cell hemoglobin S, or HbS, stick to one another, forming long polymeric chains. These long polymeric chains are locked into deoxy form and they cannot bind oxygen, leading to anemia.